Our next speaker is Gavin Dehnert, emerging contaminant scientist with Wisconsin Sea Grant, presenting virtually.
All right, that should be good to go. Hello, everyone. Thank you for coming today. Sorry I can't be there in person. Today, I'm going to talk to you a little bit about some of the work that's been going on in my lab for probably about the last 10 years or so. And we're going to kind of explore the impacts of aquatic herbicide application on non-target organisms and really kind of focus on bridging the gap between laboratory and field work. And I first want to also recognize that, some of this work is stuff that I have done. Some of this is some of the graduate students and kind of pass that on. So for those in the Midwest, when we have our summers and falls, we have these beautiful lakes, very prestige, fun to kind of go out on the lake, has some recreational purposes and everything, fishing, etc. But we have a non-native nuisance plant that is known as Eurasian watermilfoil, which is constantly being introduced to new lakes. And part of the issue with this nuisance plants is it creates these mats on the surface, as you see shown here in the picture. And those mats themselves, they kind of do a couple things that are both bad for the environment. One, they almost create an environment that's unlivable for fish and other creatures just because it becomes so dense. It takes over those the native species that are in the lake itself, so it kind of becomes a monoculture. But then on the recreational side is these mats are so bad that they actually kind of make a lake unusable. So we're not able to boat through it, etc. So one way that management agencies like the Dehnert and lake associations can do to get rid of this invasive species or non-native species, is we can use chemical herbicide applications. And one of the most common herbicide that is used in the Midwest, or at least was one of the most common, was 2,4-dichlorophenoxyacetic acid, or 2,4-D. 2,4-D has been around since the early 1940s. It's pretty much well known for use both in agricultural environments, but as well as direct application into our aquatic environments. So this is a herbicide that we are directly putting into our lakes. And it's very effective. It's efficient and effective to getting rid of Eurasian watermilfoil, as well as some of these other non-native nuisance plants. However, the impacts on these non-target organisms have kind of been up in the air, and that's been what we've been trying to solve. So back in the early 50s and 60s and 70s, there was some research that was completed on the impacts towards non-native fish species. In particular, they did the typical studies, LC50s, etc., and they found that the no observable effect level, or that NOEC, was approximately about 14.2 parts per million, depending on the species. So with that, the EPA allows the permits of 2,4-D into our aquatic environments with a maximum application rate up to 4 parts per million for spot treatment and up to 2 parts per million for an entire lake treatment. Now, we don't always get all the way to those concentrations, but that's what's permittable. Additionally, because 2,4-D is also used in agricultural scenarios, it can also enter our aquatic ecosystems through terrestrial runoff. And some of those concentrations, can get to as high as 0.7 parts per million. So those ecologically relevant concentrations that we kind of like to think about is pretty much anything below 4 parts per million from when we're directly applying it to a lake, but then, up to about 0.7 when we are including terrestrial runoff. Now, there was a recent study that kind of kicked off all of this work back in 2016 by my predecessor, where he found that these early life stages of fathead minnows had seen some impacts to survival at extremely low levels. And these levels were, at, up to 2 parts per million, but as low as 0.05 parts per million. We then decided to start to repeat this process and kind of look at, do different 2,4-D herbicides formulations, are we seeing the same impacts? We found very similar impacts, that these early life stages, they were being impacted by survival was being impacted at concentrations as low as 0.05 parts per million. Further, as we kind of went along, we did this for more species. Other labs had explored additional fish species, and we really were starting to see, regardless of which species that we were looking at, these concentrations that we were applying to the lakes were showing some impacts to those early life stages of these fish, particularly that embryo and that larval stage. Now, we did a lot of this work. We published 3 or 4 papers on this stuff, but we kept getting this question over and over again. You did this in a laboratory, so what does that mean for real life scenario? How does what we do in a lab, how does that actually translate to what we know in the environment? And so that gets to my kind of next question is like, how do we start to bridge that gap between what's done in the environment and what's done in a laboratory versus what could happen in the environment? And so the first way that we started to do this is we decided to do embryo exposure assays again. And we chose walleye because walleye were really a known sensitive species, but as well as they're a game species that people in the Midwest really enjoy to fish for, as well as eat. And without going into too much detail about the exposure assays, we would expose the embryos from fertilization through hatch to different concentrations of 2,4-D. Those concentrations were in that environmentally relevant concentrations. But to start to bridge that gap, instead of using system water like we've always done before, what we decided to do is go out to the lake itself, bring back that lake water into the lab, and therefore we were going to have different water parameters, et cetera, et cetera. And this is a busy graph, so I'll just kind of point out the 2 biggest things. Is when we're looking at graph A, we notice 2 big things. That once we increase the concentration of 2,4-D, so moving from left to right, we see a decrease in survivorship for all of the concentrations that we tested, and including at every individual lake. So the different lake parameter waters, et cetera, weren't really changing what we saw. However, I'll draw your attention to Lake Mendota here, is we see a severe decrease in the controls, but as well as all of the other treatments. And what that's really kind of pointing at is maybe there's some other things that are in the lake that can be having this having an impact on the survivorship as well. But really the same thing kind of goes that in B, when we have it normalized to, all of our control data would be 100%, we still see the very similar patterns of a decrease in survivorship. And all of this was showing that regardless of if we use different lake water and system water, which is our E3 embryo here, we see pretty similar results. And that was really encouraging to start to think. We did this using the larval stage as well, right? Those 2 sensitive stages. And again, we see very similar impacts. When they're exposed to lake water in 2,4-D, whether system water in 2,4-D, we see similar decreases in larval survival at these ecologically relevant concentrations. But the same question kept coming up. So what, you did it in a lab again? Yeah, you used different lake waters that had unique parameters, but how does that relate to real life scenarios? So we decided to take this a step further, and we decided to say, okay, what happens if we go to a lake that is undergoing an actual herbicide treatment, and we try to bring our lab experiments to those different herbicide applications. And so for 2,4-D, we were able to look at two different lakes, Dutch Hollow and Big Lake. And just to point out, the concentration that they used in that lake were 0.8 parts per million and 0.37 parts per million. So that same range that we're starting to see impacts. In the first way that we wanted to assess, actual lake herbicide on the impacts of these fish is we decided to do a mobile exposure system. So the way that this works is it's basically mimicking what we do in the lab, only we can pump water directly out of the lake into a head tank up here, which then gets distributed to each of these individual tanks that'll have different non-target organisms in them. And the second was an in-situ exposure system. And this is going back to your true bucket biology. And this one we thought would be the most representative of actual in-lake herbicide treatment, where we were able to put our non-target organisms, in this case, fathead minnows, into these buckets. And then during an actual herbicide application, you would be able to see the impacts. So what did we find? When we, for both of these lakes, compared to our control, so we did this once when there was no herbicide being exposed into the lake and once during the actual herbicide in the lake, we saw very similar decreases in survivorship, where we saw about a decrease in survivorship, about 20 to 30%, which is very similar to what we saw in the laboratory studies before. And this was similar for both Big Lake and Dutch Hollow. For the in-situ exposure system, we see pretty similar impacts as we saw with the mobile exposure system, but as well as what was in the laboratory. Only this time you kind of see much higher survivorship for our controls, but kind of similar survivorship for the treatment groups. So kind of hinting at this idea that maybe these treatment group or what we're seeing regardless of for the herbicide treatment, that 40 to 50% decrease is about what we're seeing here. This was really exciting. We were super happy to see these results. However, we were planning then to repeat this for the following year, but with the shortage of the COVID shortage of different supply chain and et cetera, 2,4-D was not used for a whole single year in the state of Wisconsin, which was great, but that didn't stop them from using herbicide treatments. And they switched to a brand new herbicide that had just been approved by the EPA called Procelicor. And they decided to kind of do some of these herbicide treatments with this new herbicide. So regardless, we were like, let's not scratch an entire year. Let's ensure that let's kind of do a little pilot study. So we had done all this research in the lab for 2,4-D. We showed that you could go from the laboratory, really matched what you would see for in the lake ecosystems. And the next kind of version with Procelicor, we were like, let's take the opposite approach. Let's see what happens with all these different Procelicor treatments and see what we can learn just as maybe a starting point. And so we looked at 5 different lakes that went underneath these herbicide treatments. We chose to focus on that in-situ exposure system, the bucket biology, because one, it was had the highest survivorship in the controls, and it was actually the least amount of effort for us. So we were able to put them in the lake, come back 21 days later and see what happened. So in short, if we just to kind of give our first findings, and we're still kind of working through this, is that when we just compare all of our control lakes compared to all of our treatment lakes, we do again see a decrease in survivorship, somewhere in that range of about 15 to 20%, which is less than what it was for 2,4-D, but still something to potentially explore. When we broke it down lake by lake, we can see that these results were really driven by two lakes, the ALC, which is Anderson Lake, and ELC, which is Elwood. And so we're still kind of figuring that stuff out. So all in all, what this kind of, the point of this was, we can use laboratory work, we can use field work, and we can kind of bring them together. Thank you, everybody. Sorry, I know I'm running out of time. And maybe potentially take any questions.
Unfortunately, we are out of time for questions, but you can continue the conversation on HUBA. Thank you.